Our paper reviews and discusses the current challenges to enhance the physiological relevance of human tissue models. Flat 2D cultures are the current standards, but we all know that life is not in 2D, so it's necessary to develop 3D in vitro models. This figure illustrates the key message of our paper, which is that the existing paradigm of 3D culture suffers from a lack of control over organoid size and shape. This can be an obstacle for cell harvesting, and secretions cannot be collected from organoids embedded in a gel, making cell function analysis by multi-omics approaches very difficult. The other aspect of our key message is the need to create 3D cell culture systems that better mimic real gland structures, which look a little bit like grape. The advantage of creating this grape-like structure is that we can collect the secretions from the glands for further analysis. Here we see a typical exocrine architecture that we can find in prostate, breast, salivary, pancreatic glands, etc. These glands consist of ducts that collect secretions from acini. This scheme illustrates the complex structure of an acinus when cells self-organize into a gland. This glandular structure can be recreated in a gel-based 3D culture that entraps all the microenvironmental components inside the gel. But the problem is that secretions cannot be assessed. To allow access to luminal secretion, a wide range of scaffolds has been created in the last decades to support 3D cell culture. This set of scaffolds includes open and closed microtubes with polyelectrolyte shell, microtubes lined with prostatic epithelial cells and with mammary cells, 3D scaffolds designed to mimic human intestinal villi, hemisphere-like structures, microbead-based culture, microcarrier-based culture, and microencapsulated 3D co-cultures. These different components of the 3D toolbox can be combined depending on the application. For example, if we need to perform flow-based drug screening, we would use microfluidics. If we need to perform secretomics-based diagnosis, we would combine acinar-like structures with duct-like structures. This bio-inspired approach to combine cell culture with microfabrication techniques provides us with a new generation of tools for multi-omics analysis.